Well, first off, I want to congratulate you on completing your pharmacokinetic study,
Yep.
and that's
Thank
been long coming. I've been kind of watching you for the past decade, so this
you.
has been long in the works. Um,
Thanks.
if we could, why don't you give us a little background on what you guys learned and what ground you guys have gained and
Sure.
where this may take us.
Okay, great. So the first thing that I want to do is I want to make clear my financial disclosures. So as you know, in 2004 we discovered the eukaryotic nicotinamide riboside kinase pathway. We discovered that NR is a vitamin in everything from a yeast cell to a human being. That intellectual property from Dartmouth was licensed by Chromadex Corporation, which has developed nicotinamide riboside as a GRAS and GMP produced ingredient. And so I have an IP interest in nicotinamide riboside. I'm on the scientific advisory board of Chromadex. And then in addition to sort of newer development is that I'm a co-founder and the chief scientific officer of a company called Pro Health Span. And this is the product that we have at Pro Health Span, True Niagen. It's available at prohealthspan.com. And we're also both at Chromadex and at Pro Health Span. We are trying to build the knowledge base about the science of nicotinamide riboside so that people like yourself in your community that are interested in taking it can have up-to-date, scientifically sound, peer-reviewed information about NR.
So, my questions are going to be coming from the user base that you helped grow. I'm one of them. I've been taking it since January of 2014.
Great.
I'm the curator of the nicotinamide riboside group on Long City, and I'm taking about a gram a day, and many other users are up around my level as well.
A gram a day. Okay. And so you're taking 825 milligram capsules.
Yes.
And are you taking all 8 in the morning?
No, I spread them out through the day.
Mm-hmm.
After some of your initial research, that was the direction I went with.
Okay, so I think that we should really start with an understanding of, what's the purpose of taking NR and what are the effects of NR and when do the effects of NR peak? When you get a peak NAD after supplementation, which is around 8 hours, and the fact that we're taking NR in order to increase NAD availability and the availability of other NAD metabolites, right? And we were actually the first group to show in 2007 that nicotinamide riboside increases the activity of SIR2, right? So if you want to think about NR as a stack, right, as a sirtuin activating compound, it does that. We demonstrated that in 2007 that we could increase yeast lifespan and SIR2 activity with nicotinamide riboside. When you're increasing NAD formation, when you're boosting NAD formation with NR, you're increasing NAD consumption by sirtuins. That's the whole point, right? So you don't necessarily have to see an increase in NAD plus in every tissue to have an increase in NAD metabolism. To have either an observation of the whole NAD metabolome, which we do routinely. I think we're still the only group in the world that does that routinely and quantitatively, or you need a flux experiment in which you're tracking, the nicotinamide moiety and the ribose moiety, and you're seeing what it's being turned into. But the fact that NAD peaks in 8 hours doesn't mean that you have to take NR every 4 hours or every 8 hours. It doesn't follow to me that you would necessarily want to do that. The body has a circadian cycle of NAD metabolism. In a mouse, there are two NAD peaks per day. One in the daytime, which is similar to our nighttime, and one in the nighttime, which is largely driven by food consumption. Essentially, so when I take NR at, when I rise up, I'm boosting my daytime NAD peak without increasing my calorie consumption, right? If I were trying to boost my day and nighttime peak, I would split a daily dose into, first thing in the morning dose and an evening dose. But I don't see any point in splitting it more than 2 times per day. That doesn't make any sense to me.
Alrighty, so that clears up a lot. So is there anything to consider in terms of taking it on an empty stomach or a full stomach?
I take it on an empty stomach, but I'm about to eat food, so I don't think it's a real concern either way.
Is there anything food-wise that would enhance its absorption?
That's a good question, and I don't think it's been really studied. We do all of our mouse and rat experiments by mixing. We either mix NR into food, into mouse chow, or we deliver it by oral gavage, which is basically dropping it into the stomach. There are some people that put it into the drinking water, but it's a very water-soluble compound. It's not going to go into a oil phase, but it could be that there are certain types of food that would enhance its availability, but I don't think that anybody knows.
There have been some questions about methylation, and that maybe we should take folate or something like that in addition to NR. Are we using up any nutrients that we need to replace if we're boosting NAD?
Possible that at really high levels of supplementation you would affect methyl group metabolism, but I don't think there's a quantitative answer to that. It's folate. There's a rationale for taking folate, but quantitatively, I don't think there are sufficient data one way or another. So it's been a really exciting 12-year period. Just 12 years ago, we had the first evidence of NR from basically a petri dish with yeast colonies growing on it. And today, just 12 years later, we estimate there are about 200,000 people that are taking NR daily. Typically 250 milligrams a day. And there have been many dozens of preclinical studies, largely in mice, but additionally in worms. We have one coming out in rats, and I'm sure there's other model organisms that are showing very remarkable metabolism boosting activities of nicotinamide riboside. And so it's critically important that we get some human clinical data that shows its oral availability and allows us the quantitative tools to measure its activity and the time course by which doses of NR boost NAD metabolism. And ultimately, we want to move into efficacy trials so that, as NR is marketed as an over-the-counter supplement, right? So it's not intended to treat or prevent any disease or condition. The suspicion is, however, that people with diseases and conditions may may enjoy even greater benefits than healthy people, because when we test NR in metabolically stressed animals, we see really remarkable improvements in things like insulin sensitivity, weight control, prevention of diabetic and chemotherapeutic neuropathy, protection against noise-induced hearing loss, and the like. So going forward, there are going to be tests not only of the bioavailability and safety of nicotinamide riboside, but also the efficacy of NR in doing a lot of the things that we think it will do, including improve plasma lipids, potentially increase HDL, lower LDL, improve weight management, and protection against nerve damage, as I was saying.
There is a theme I've kind of picked up from your papers and some of the other researchers out there, that this is a shared metabolite, that cells share this in states of injury, in times of disease. Healthy tissue seems to lend a hand with tissue that needs NAD. Is this coming through some of the research?
Yeah. So let's talk about what happens when a human being or a mouse takes NR. And we deliver NR orally. In all of our mouse or rat experiments, we're either mixing NR into the food of a mouse or rat, or we're delivering it into the stomach by oral gavage. And so it's, very similar to a human being taking a couple capsules of NR, in the morning. And one of the things that you see in a mouse is that the liver NAD pool expands really remarkably after oral ingestion of nicotinamide riboside. We also know that blood NAD rises pretty significantly with oral nicotinamide riboside, so up to 2.7-fold about 8 hours after oral ingestion of a gram of nicotinamide riboside. Blood NAD can be up 2.7-fold. Now, when tissues and blood cells take up NR, they're boosting not only NAD+, but other critical NAD metabolites like NADPH, which is crucial and limiting for resistance to reactive oxygen species, the ability to make steroid hormones like androgens and estrogen, and other biomarker metabolites. As we recently described, NAAD was unexpectedly, strikingly boosted by oral NR, and that's a good way of telling that NAD metabolism is increased. But one of the reasons that we supplement with NR is to increase the activity of sirtuins, right?
Right.
So when you increase NAD+, you make more NAD+ available to the sirtuins, and sirtuin activity is going to convert NAD to nicotinamide and what we call an ADP ribosyl product, typically acetylated ADP ribose and acylated ADP ribose, and those metabolites get further processed. So the liver can expand its NAD pool, and then after the liver increases sirtuin function, the liver has more nicotinamide available to it, and the liver can sustain an increase in NAD metabolism with that released nicotinamide, or it can release that nicotinamide to other tissues. And so when we take NR, we're boosting NAD in a first wave to a number of tissues through the nicotinamide riboside pathway, and we're making NAD metabolites, principally nicotinamide, available to additional tissues. Um, really high dose nicotinamide, orally high dose nicotinamide doesn't strikingly increase the activity of sirtuins in the liver, and we recently demonstrated that. But we think that the secondary nicotinamide that is more slowly released from sirtuin activity can generally increase NAD metabolism. So there's a couple of waves of increase in NAD metabolism that comes from, as you say, circulating or sharing non-phosphorylated NAD precursors. The three NAD precursor vitamins are NR, nicotinamide, and nicotinic acid. You'll see that there are some researchers out there that use NMN and that have tried to boost and succeeded really in boosting mouse NAD metabolism with a metabolite called NMN, which is actually the product of the nicotinamide riboside kinase reaction. So NR gets phosphorylated with ATP to NMN. Similarly, nicotinamide gets phosphoribosylated, fancy word, to NMN. And so, and NAMPT, the enzyme that does that reaction, declines in aging, right? And so some people thought that by boosting NAD metabolism with NMN, you would bypass NAMPT, and you'd even bypass NRK, and you'd make the product of the NAMPT or NRK reaction. So we had two papers come out this last week. On Monday, we had our paper on the first clinical study of nicotinamide riboside, and on Tuesday, we had a paper that was collaborative with Carlos Canto from Nestle and in Switzerland, and he made a knockout mouse of NRK1. And with that knockout mouse, we showed that both nicotinamide riboside and NMN depend upon the nicotinamide riboside kinase pathway to boost intracellular NAD. So essentially, NMN gets dephosphorylated, gets converted into NR.
That was a point I was hoping you would make, because some of us have begun to look into that, and it seems like we're already there with NR. It's we're getting it direct, whereas if we had availability of NMN, it would still get converted to NR and then back.
That's the thing. So compounds that have a phosphate don't get transported into cells, and so we actually think that there's a role for circulating NAD, and that circulating NAD and circulating NMN can indirectly boost intracellular NAD metabolism, but it's not by envelopment or import of intact nucleotides, NAD, NMN. It's rather by conversion to NR or nicotinamide that those compounds can boost intracellular NAD metabolism. Hang on one second. So we showed that that NMN is extracellularly dephosphorylated to NR, and you knock out NRK1, and neither nicotinamide riboside nor nicotinamide mononucleotide can effectively boost NAD metabolism. So NMN, even though you think it might be a step after NRK, actually is itself a precursor to extracellular NR, which then goes into a cell and becomes NMN and NAD inside the cell.
So the transport is across, what is it, the PNC transporters?
No, so PNC is a bacterial enzyme PNC1 is actually nicotinamidase. So PNC1 is and in terms of vertebrate biology, so we're vertebrates, we have a backbone, right? So, so vertebrates do not encode nicotinamidase, but you know about the microbiome, right? So our gut contains a tremendous population of bacteria, many different kinds of bacteria.
Right.
In our gut, nicotinamide can be converted to nicotinic acid. So that's what PNC does. Now, the transporter for
You're talking at the cell membrane for us.
the transporter for NR is basically a nucleoside transporter, NT, and it's pretty generic in humans and other vertebrates, meaning that it will transport adenosine, cytosine, NR. It's not specific. In yeast, one of my best grad students ever, Peter Belenke, discovered the yeast nicotinamide riboside transporter, which we called NRT1. But the thing about molecular biology is it only takes you 5 minutes to know whether there's a human homolog, a human sequence homolog of a yeast gene, because you basically put the sequence into a computer, into a browser. You click the enter key, and you have a very rapid search of human genomes, and you can determine whether or not there is a sequence and structural homolog of a yeast gene or a bacterial gene. And so we've known for a long time that there isn't something that looks like a dedicated NR transporter, but we do know pharmacologically that NR depends upon regular vanilla garden variety nucleoside transporters, and there's a group out there that is searching for an NMN transporter, but we don't think there is such a thing. And our recent paper in Nature Communications shows, as I said, that NMN intracellular availability depends on NRK1, so it's inconsistent with the idea that NMN is transported.
Well, there was an article I picked up on some time ago. It was lower limb muscle injury enables quantitative classification of healing, and what they showed in that article is an increase in NMRK2. I don't know if you read that article.
Yeah.
So, is that something along the lines of this cell-to-cell sharing? Is there available NAD precursors, and this increases in the cells to help this exchange?
Brian, it's a really good question. So here's a typical. This doesn't happen every week, but probably about 3 or 4 times a year, I get a phone call or an email from somebody saying, I'm looking at such and such a disease model. It's usually something muscular. It's usually cardiac, the heart muscle, right, or skeletal muscle. Someone will say, I've got a model of dilated cardiomyopathy or some kind of skeletal muscle or nerve damage, and I did a microarray, so I did a gene expression profile, right? And they tell me the most highly induced gene is NRK2. Sometimes there's an M in there. In the human gene symbol, it's NMRK2. We don't always pronounce the M. So it's nicotinamide riboside kinase 2. And so there's 2 implications of that. One is that if we were to provide that injured animal with nicotinamide riboside, we could boost NAD metabolism in that damaged tissue and very possibly ameliorate the disease or condition that is leading to gene induction of NRK2. And we found that in a number of cases. And secondly, why would the damaged tissue, which is I think where you're going with this question, why would the damaged tissue be inducing NRK2 unless there's a source of nicotinamide riboside to that tissue, right? So And I'm a good person to ask that question because A, I clone nicotinamide riboside kinase. Two, any condition. So,
You might have to repeat that last sentence. I lost you here at this end for a moment.
sorry. You may know that we developed quantitative targeted NAD metabolomics, which means we use mass spec to quantify the levels of essentially all of the metabolites in the NAD metabolome: NAD, NADP, NAAD, nicotinamide, and NR, and so forth. And the problem is, and it's quite frustrating, is that in blood, it's very difficult to detect NR. Even in conditions in which we know there's circulating NR, NR appears to be cell-associated, difficult to extract from blood, and difficult, therefore, to quantify. But we think that there must be probably some localized cell lysis that would make NR available. And let me bring in another thought that I think may be useful to the readers of the website, and that is, where does NR come from in nature, right? And everyone on your website is going to say milk, right? Because we published that in 2004, and earlier this year we quantified that in milk is around 5 micromolar. So you'd have to drink hundreds or thousands of glasses of milk in order to get the hundreds of milligrams of NR that that people are boosting with. But if you were to ask me what is the real source of NR in our diet, I would tell you non-processed foods. I would tell you whole foods with a small w and a small f. But it's not chemically NR. It's largely in the form of NAD+. So the most abundant NAD metabolites are NAD, NADPH, NADP, NADH, right? Followed by everything else. And in the course of digestion, our enzymes and bacteria are converting those phosphorylated metabolites to the non-phosphorylated NAD precursor vitamins, namely NR and nicotinamide nicotinic acid. The least processing of NAD will give you NR, which is going to go through nucleoside transport, as we said, to go through the NRK pathway and consume the least amount of ATP in order to be salvaged to NAD+. And then cleavage of the glycosidic linkage between the nicotinamide moiety and the ribose moiety is going to yield nicotinamide. That can go through the NAMPT pathway. And then if nicotinamide is deamidated by this bacterial product, PNC, to nicotinic acid, then that can be used to regenerate NAD in the tissues that express that pathway, nicotinic acid, phosphoribosyltransferase transferase. So the real source of NR is food, right? It's digestion of food. But we think that both as we age and under conditions of cellular stress, there's a need for higher levels of nicotinamide riboside. So if you're supplementing with NR, you can get through those stresses of life. But in the ailing heart, exactly where the NR would be mobilized from is not really known, and it's a very interesting question. It's something that we're quite interested in solving. I suspect it's from lysing cells.
I have read a number of articles on this. They have produced states of ischemia in brain tissue, heart tissue, for NAD experiments. Is anyone approaching that on the NR side to see what advantages we could have with that?
Yeah, I mean, you should see my email inbox with with papers to review from really all around the world. There's a tremendous amount of stuff that's going on in almost any disease model that you can imagine. NAD is being so central to cellular bioenergetics, DNA repair, resistance to reactive oxygen species damage, steroid synthesis, you name it. Twins, calcium signaling in production of, cyclic ATP ribose and those types of metabolites. So there's an incredible intersection between NR and all sorts of disease states.
So that kind of answers my question a little bit on the NMRK expression. So in states of tissue damage, we will see elevated levels where we might not expect them in certain tissues, right?
Yeah, so one of the valuable things about NR as a particularly valuable NAD precursor vitamin is the NRK1 enzyme is constitutive. It's expressed in virtually every cell that we know of. And it doesn't seem to decline in aging. It's just there. Nicotinamide Riboside kinase 2 is more muscle and nerve specific and then is induced by damage. So the gene expression pathways, or the genes that are responsible for converting NR to NAD, have some advantages in that one or both of those enzymes is expressed in every cell. And then, as you know, high-dose nicotinamide inhibits the sirtuins, whereas high-dose NR doesn't. And high-dose nicotinic acid, although has some very exciting and valuable properties like elevating HDL and lowering LDL, its use is limited by flushing. And you know yourself, if you're taking a gram of NR a day, that you're not experiencing a flush response. Am I right?
No, not at all. Not at all. Could you comment at all maybe on the epigenetic side of this and how NR may help towards maintaining a more youthful profile of our genome?
Yeah, so some of the sirtuins, namely SIRT1, is heavily engaged in chromatin modification reactions. And the founding member of the sirtuin family, yeast SIR2, is a protein lysine deacetylase that specializes in deacetylating sites on histone H3 and histone H4. It controls a number of interesting genes in yeast, including the mating type locus. But I think the key to longevity in the yeast system has to do with deacetylating a particular site on the telomeric structures on yeast chromosomes. And so these are the ends of chromosomes, and chromosome ends can shorten if they're not maintained properly. And so by providing a high enough level of NAD, the sirtuins stay active, and the sirtuins have a number of different targets. Some of them are on chromatin and regulate gene expression at the transcriptional level. There are a number of metabolic targets as well. SIRT3, 4, and 5 are the mitochondrial sirtuins, and they're not so much involved in regulation of gene transcription, but really regulating gene activity at the level of enzyme function. So under a number of conditions, mitochondrial proteins become inhibited by virtue of protein lysine residues being modified by acyl groups, acetyl, propionyl, and other acyl groups. And the mitochondrial sirtuins remove those modifications and they kind of boost mitochondrial metabolism by cleaning up those enzymes and getting them to work in a form that's sort of as good as new.
So it's very important in terms of maintaining what did you call the where the DNA wraps around those molecules?
So yeah, histones are the most abundant chromosomal proteins in the nucleosome.
Mm-hmm.
There's 8 copies of histones in every nucleosome, and there's a number of modifications on those histones that control gene expression, and sirtuins, in particularly human SIRT1, has a role in regulating those nuclear modifications, those nuclear histone marks.
And these are all NAD-dependent actions.
Sirtuin-mediated deacetylation is NAD dependent. There are other deacetylases that don't require NAD, but the sirtuins have an absolute requirement for NAD.
There has been some talk, and this is from one of the longevity groups out there, that we may be overworking our old mitochondria by stimulating all this extra NAD production. I'd like you to comment on the biogenesis portion of our mitochondria and how your body, your cell sorts out the good and the bad.
Right. So you need mitochondria for most ATP production in oxygenated tissues. You need mitochondria for gluconeogenesis, because to get from a compound called pyruvate to oxaloacetate, you need mitochondrial reactions. You need mitochondria to deal with reactive oxygen species, to do quite a bit of amino acid oxidation. Almost every tissue has critical roles for mitochondria. And mitochondria are kind of a fascinating organelle. They have their own genome, a very small one, that largely encodes the electron transport chain. The rest of the mitochondrial proteins are actually encoded from the nuclear genome and are imported to mitochondria. So there's mitochondrial protein quality control, and mitochondrial protein turnover is important for mitochondrial function. There's also something called mitophagy, where whole mitochondria can be engulfed and turned over and recycled. As far as we can tell, mitochondrial turnover is essential for health span, but I don't think that generating new mitochondria is a cellular stress. It seems to me it's a homeostatic health promoting response.
So there's no reason to fear boosting our NAD, that our aging mitochondria can certainly handle this.
Well, when you're boosting NAD, you're boosting nucleocytosolic NAD as well as mitochondrial NAD, and there's a balance. And there's a lot of things that cells know how to do sort of automatically, and converting NAD precursors to NAD plus and NADP is something that cells are programmed to do, and it's not. It actually takes demonstrably less ATP to form NAD from NR than from any other precursor. So it takes much more ATP to produce NAD from tryptophan, nicotinic acid, or nicotinamide than from NR. And so that's one of the reasons we think that when a heart cell is in failure mode, it induces expression of NRK2, because conversion of NR to NAD would stress the heart the least. So a failing heart has low phosphocreatine and low ATP, and so it potentially, quote, wants, if a heart cell wants anything, or anthropomorphizing there. It would want the NAD precursor that would allow it to restore its NAD with the least amount of ATP input, and that's through the NRK pathway. The thing is, it's early days, and there's still a whole lot more that we've done in animal research than we've done in humans, right? And so, I think that what we need is more evidence basis, right? We need placebo-controlled trials. And I'm very pleased to tell you that on the basis of very promising experiments done in the rat, in a collaboration with Donna Hammond at the University of Iowa, in which we're showing that NR allows a rat to resist chemotherapy-induced peripheral neuropathy, that we've been funded by the National Cancer Institute to conduct a human clinical trial in women undergoing cancer chemotherapy to see whether they'll be able to complete their rounds of chemo without suffering from neuropathy. So we really need more human data to rationalize the uses, particularly for diseases and conditions.
Well, maybe since you touched on that, on cancers, maybe I can ask you this. Since that Australian clinical trial came out suggesting that a B3 derivative could cut new skin cancers, how would this be relevant to us with NR?
So probably that's boosting the NAD that is used by PARP1, which is a critical mediator of single-strand DNA break repair. Skin cancers largely derive from sun-damaged DNA. So the sun is a mutagen, and the repair process requires a supply of NAD in order to signal the presence of damaged DNA and assemble a set of enzymes that do the repair. And when too much NAD is turned over to generate that damage signal, then NAD is not available for other cells, and then you get not only damaged DNA, but a damaged cell that can be a precursor to malignancy. So nicotinic acid, I think, is probably what they used in that trial. But it will be very interesting to see the degree to which NR is protective.
It was nicotinamide is what they had used.
Nicotinamide, okay.
Yeah. And it was phase 3 clinical trial, so this has been ongoing for a little while.
Great.
Well, maybe you can take us full circle and land this plane for us. Maybe you can tell us what was previously known about B3 benefits with arthritis, memory, why it's taken so long to bring this back to these discoveries, because here we are for the last 70 years. A lot of these benefits were known. Why has it taken us so long to get back here?
It's funny, micronutrient research isn't always fully appreciated in mainstream biomedicine, and but you're absolutely right that there have been some strong proponents of high dose niacin in psychiatric conditions, and certainly in dyslipidemia for a long time. And I occasionally run into people that use niacin for additional things I'd never heard of, like getting to sleep. And so I think that we really need more placebo-controlled trials to investigate the activities of NR in a number of the things that nicotinic acid has been used for. And because I think that we have in NR a compound that is safe, that is going to be available to more tissues than nicotinic acid is, and is not going to be complicated by the issue of flushing. So I'm really excited about the future of NR research, and in particular doing placebo-controlled trials in a number of the conditions that you mentioned.
Well, I think we're all very excited about that, and I think at the moment there's 10 registered human clinical trials. 4, I believe, have been completed. Yours is really the first publication that's come out on this so far. We're all just very excited at this end, and we're all listening to whatever you guys can turn up for us.
Great. Well, thanks for being in touch, and you can definitely follow the ProHealthSpan website and the ChromaDex website for further information. My university website is biochem.uiowa.edu/Brenner, and good luck, and I hope that we are able to communicate in the future when we have further publications.